Have you ever wondered how the time it took to recover from a common cold, the flu, or other infections has gotten shorter after you were exposed to and successfully recovered the first time?
You're sneezing!
This is due to acquired immunity, the work of the adaptive immune system, which uses specific antigens to work up an immune response. This is different from the innate immune system, which attacks only based on the identification of general threats, uses macrophages, neutrophils, natural killer cells, dendritic cells, basophils, and eosinophils, has the key components of antimicrobial peptides and proteins, and does not have any immunological memory.
Adaptive or acquired immunity, on the other hand, is activated by exposure to pathogens and uses an immunological memory to learn about the threat and enhance the immune response. The adaptive immune system relies on fewer types of cells, T cells, B cells, and other antigen-presenting cells, than the innate immune system to perform its functions.
These cells are B cells and T cells, both of which are lymphocytes that are derived from multipotent hematopoietic stem cells found in the bone marrow. After being in the bone marrow, these cells mature and become activated, each following a different path to their final mature forms.
After maturation in the bone marrow, the B cells move into the lymphatic system to circulate throughout the body. Here, the B cell comes into contact with an antigen, which kickstarts the maturation process for the B cell. But this antigen has to fit the B cell's antigen-specific receptor on its surface that is inherent to the organism's DNA. B cells have antibodies on their cell surface called membrane-bound antibodies. Once the B cell encounters the antigen that matches its membrane-bound antibody, it divides in a process known as differentiation.
This process can also be initiated by a helper T cell because B cells are also antigen-presenting cells that chew up an antigen and present it in the form of an MHC II complex. In the process of differentiation, B cells become either a memory B cell or an effector B cell, also known as a plasma cell. Unlike memory B cells, plasma B cells aren't membrane-bound and can secrete antibodies. B cells can also express a specialized receptor called the B cell receptor, which assists with antigen binding. B cell receptors can also initiate signaling pathways such as cytokine release, serving to communicate with other cells of the immune system.
After being formed in the bone marrow, T progenitor cells move into the thymus to become T cells. While there, the cells begin to express T cell receptors and other receptors called CD4 and CD8. T cells don't have antibodies that can bind to antigens. Instead, T cell receptors can only recognize antigens that are bound to receptor molecules called major histocompatibility complex class 1, or MHC1, and class 2, MHC2, as seen with the B cell differentiation, which could be activated by a helper T cell attaching to an MHC2.
These MHC molecules are membrane-bound surface receptors on antigen-presenting cells, such as dendritic cells and macrophages. CD4 and CD8 bind to either MH1 or MH2 for T cell recognition and activation. CD4 positive T cells are referred to as helper T cells and are attracted to the MHC2 complex. CD8 positive T cells are attracted to MHC1 complex and cytotoxic. CD8 kills bad cells such as cancerous cells or virus-infected cells like B cells. T cells differentiate into effector and memory cells. An effector helper T cell releases cytokines that act as an alarm to other cells to get in gear for an infection in the body. Cytotoxic T cells and their effector cells kill cells.